Good morning. Today I'm going to show you how to extract DNA from pea cells. Well, we're actually working with red lentils because I think they're just kind of a fun color. So the first thing we're going to do is we're going to get all our ingredients together, which I've already done for you. I like using distilled water. You don't have to. You can use tap water. I've got just plain salt, meat tenderizer, 90% isopropyl alcohol, or just a rubbing alcohol you would find at your drugstore. Some clear liquid, either dish soap or hand soap, either will work, and you can use other colors, they're fine, just sometimes they tint your results a little bit. And that's it. So the first thing we're going to do is we're going to take half a cup of our red lentils and put them in a blender. And in that blender, we're going to put a 1/4 teaspoon of salt, and I am such a precise measurementer. Is that a word? I don't think it's a word. Anyways, I put in a little bit of salt, and then I need a cup of water. So I'm going to use my distilled water to measure to a cup. Now remember, when you're measuring, this doesn't have to be accurate, precise, but you always want to get down to the height that you're measuring at and look at the bottom of your meniscus where it dips down to make sure it's right on that line you're measuring to. You're going to add that to your cup. Half a cup, and then you're going to put it in the blender and blend it until it's the consistency of like a real thin pea soup. Hold your ears for a second, it's going to be kind of loud. I'm going to look at it just to see. Well, interesting smell. All right, I'm almost there. I want to get it a little bit more just because this is the mechanical breakdown of the peas because we're trying to get to their cellular level. So we're going to break it down a little bit more with our mechanical. I'm going to take my pea soup solution, I'm going to pour it through any kind of sieve, or you can use filters. Pour it through, and I'm going to strain it because I just want the liquid coming through, not all the cells. And this is going to take a little while to go through something this fine. All right, I got a spoon to help me get it through here. I'm going to stir it to help it get through a little bit faster because I have a really fine flour sifter here. To strain it, it's going to take a while, and if I just stir it with a spoon, it goes a little faster. Because I'm really only looking for enough liquid to fill this glass. Now, DNA is the blueprint for life. It stands for deoxyribonucleic acid, and it's composed of strands of a sugar, deoxyribose, and nucleic acid bases, and it has a phosphate group attached. We're trying to basically physically and chemically break up all those little parts so that we can get just a strand of DNA. DNA is so long, it would never fit in our cells if it was just left to itself. So cells have their DNA very, very tightly compacted and wound. They wind them on proteins called histones, and they're all tightly packed into those cells inside the nucleus, kind of like a reference library. It's going to hold all those blueprints and not let them out. So we're going to try and break them free. We're jailbreaking our DNA so we can take a look at it from a macroscopic level. You don't have to worry about getting every tiny little stitch of it. This is plenty. Kind of messy, but there you go. We've got the solids out, and we've got kind of a thinner liquid that's hopefully going to have enough cells in it that we can get some DNA out. This next part is going to help dissolve the fatty part of the cells, which is our cellular membranes, are made of phospholipids in a bilayer, where the fats kind of have their tails hiding into each other and they have phosphate groups on the outside. Soap works to dissolve and push apart fats. So you can put 2 tablespoons of a detergent into your glass, and this is simply to dissolve those membranes. Just like you would take a greasy stain out of your pants with a dish detergent or any kind of laundry detergent and kind of gently and rub it in. In this case, we're going to gently swirl in our detergent, and it will break those cellular membranes up and free the contents of the cell into the liquid. All right, I'm going to swirl this for a little bit, and then I'm going to have to let it sit for 10 minutes. Well, at least 10 minutes. You can leave it sit longer if you want. All right, looks pretty mixed to me. So I'm going to set a timer and let that sit for 10 minutes. So we've let this sit for well over 10 minutes. And I just want to scoop the foam off the top so I have a bit of room to work with here. I'm going to scoop that foam out of there. Then I'm going to add a 1/4 teaspoon of meat tenderizer. Now the meat tenderizer itself needs to be nice and fresh. It's going to release and digest those proteins that are holding the DNA tightly together so that it releases it into looser strands. You want to do this very carefully. You want to just carefully put it in and very, very gently stir, because we don't want to destroy anything we want to look at. We're simply going to get that tenderizer to interact with the proteins, dissolve them, and release that DNA. Just very, very gently mix it in. There we go. You'll notice that everything in our cup is kind of separated into layers. A lot of the more solid components of our cell are at the bottom, and then we have kind of more liquid and lighter stuff towards the top. We're going to use that separation to bring our DNA to the top. All right, so I've mixed that meat tenderizer completely in there, just very gently stirring it, and I'm actually going to pour this into a taller glass as a mixture so that I can go 50-50 and let you see what's going on as well as I can see it. So here we have this. We're going to gently pour our alcohol. It works better the higher percentage you have. I have 91%, which was the highest I could buy at the store. I'm going to gently kind of pour it on the top, and it's going to bring my DNA to the surface for me to look at. alcohol has a lower density than the rest of the cellular components, so if I'm careful in pouring this, it will sit on top of my cell solution and not actually mix with it. And the DNA will come into solution. Give it a second to sit there. We'll do it in the other one as well. You'll see these shimmering kind of strands that are in the alcohol, and that is your strands of DNA coming. Want to zoom in on those? You can see kind of these shimmering little strands in the alcohol solution. If you have a microscope at home, you could take these little guys out and just kind of wind them on a toothpick and then put them under a microscope or a magnifying glass to examine in closer detail. Here's a really good one here. Still kind of got some goop on it, but take that off and put it under your 3D microscope, and you'll get a great view of what the macroscopic outside structure of DNA is.